Okay. Okay, so basically in this video we are going to discuss for chapter 1, which is on macromolecules. For part B, structured question, so we are going to discuss first for question number 1. Okay, so question A, it says here, figure 5 shows two types of nucleic acid that can be found in living organism. Okay, so there are two basically, nucleic acid, which is DNA and also RNA. So as you know, DNA are found in the nucleus, okay, where the function of the DNA is to store genetic information, okay, and basically the size of the DNA is really large, where the genetic information are stored in the form of genes. Okay, so these genes will code for the synthesis of a specific protein. Okay, so but the synthesis of the protein will occur in the cytoplasm. So this information, okay, which is stored in the large molecule of DNA in the nucleus, has to be copied, okay, into a smaller form, which is in the form of RNA. Okay, so let's say for example, this segment here is of DNA molecule codes for gene 1. Okay, and then the second segment here is for gene 2, and then the third segment here is for gene 3. Okay, so let's say for example gene 2 code for the synthesis of enzyme 2. Okay, whatever enzyme it may be, so enzyme 2 for example. Okay, so the information in gene 2 has to be used, okay, to synthesize enzyme 2, but the synthesis of enzyme 2 occurs in the cytoplasm. So the information from gene 2 has to be copied, okay, into a smaller version, okay, into a smaller version, and this information is copied into another molecule. We call it as RNA. Okay, so this RNA, basically there are three types, which is mRNA. It stands for messenger RNA, which copies information from the DNA from gene 2. Okay, and then there is another type of RNA, which is, we call it as tRNA, which is transfer RNA. So and the third one is rRNA. Basically all these RNAs are produced in the nucleus, okay, to be specific, in the nucleolus within the nucleus. Okay, so as these RNAs are synthesized in the nucleus, they will migrate into the cytoplasm to carry out their function, which they involve in the synthesis of protein. Basically, the tRNA will involve in the to transfer specific amino acid to according to the information that is coded in the mRNA. Okay, so for example here you have a cell, okay, and then within the cell you have the nucleus. Okay, so in the nucleus you have the large DNA molecule. Okay, so from the DNA molecule it synthesizes the mRNA. So the mRNA will go to the cytoplasm, where in the cytoplasm it will find a ribosome. Okay, so as you know, ribosome is an organelle which is involved in the synthesis of protein. Okay, so the mRNA will enter into the ribosome. Okay, and then in the cytoplasm of cell, there is abundant of amino acid. So this amino acid will be carried by tRNA, okay, to the ribosome, okay, according to the according to the code which is coded in the mRNA to synthesize specific polypeptide chain. Okay, so this polypeptide chain later will become a functional protein, and in this case, if the gene is gene 2, is to synthesize enzyme 2, for example. Okay, and then another RNA is ribosomal RNA. So ribosomal RNA, okay, is the component used to is the component of ribosome. Okay, basically a ribosome is composed of RNA and also protein. Okay, alright. Okay, so that is just a basic understanding of DNA and RNA. Okay, so now we are going to look at the question. Question A shows you figure 5 shows two types of nucleic acid that can be found in living organism. Okay, so which is DNA and RNA. DNA stands for deoxyribonucleic acid, and as for RNA, it stands for ribonucleic acid. Alright. Okay, the question asks, name the monomer of nucleic acid. The monomer for nucleic acid is really easy, which is obviously it is the nucleotide. Okay, so nucleotides, you have to know what is the component of nucleotide, which is nucleotide. It is consists of three main components, which is phosphate that is bonded to a 5-carbon sugar, and then the 5-carbon sugar is bonded to a base. Okay, so for DNA, okay, the 5-carbon sugar is ribose, so that's why we call it as ribonucleic acid. Okay, and if the nucleotide is are found in DNA, so the 5-carbon sugar will be the deoxyribose. Okay, so and then you have to know that the different types of bases. So you have adenine, cytosine, guanine, and thymine. So adenine, guanine, cytosine, and thymine are found in DNA. And as for RNA, it also involves 4 nucleotides, which is adenine, guanine, cytosine, and uracil. Okay. And then another thing that you have to take note is that you have to know how to number the carbon. Okay, so this is, let's say this is carbon, the 5-carbon sugar for ribose. Okay, so you have oxygen here, and then this is carbon number 1, carbon number 2, carbon number 3, number 4, and number 5. Right, and then at carbon number 3 there is OH. Okay. Alright, question number 2, it says here, if 15% of bases in a particular DNA molecule is thymine, what is the percentage of cytosine in the same molecule? Okay, show your calculation. Alright, so you have to imagine, okay, altogether there is 100% nucleotides, okay, that consists of adenine, guanine, cytosine, and thymine. In the question, it says, okay, the percentage of thymine will be 15%. Okay, so it asks you what is the percentage of cytosine. All right, so you have to know the complementary base pairing. Okay, where in the double-stranded DNA molecule. Okay, so suppose this is double-stranded DNA molecule, and then one end we label it as 5 prime, and the other end, the complementary strand, it has to be labeled as 3 prime. Okay, so on this strand it has to end with a 5 prime that will be complement to the 5 prime here. Okay, so always remember 5 prime has to complement with 3 prime on the on the two DNA strand. Okay, so let's say, for example, okay, on the first strand here you have adenine. Okay, adenine has to pair with thymine, guanine has to pair with cytosine. Okay, cytosine has to pair with guanine, thymine has to pair with adenine. Okay, so obviously if you have 15% thymine, okay, adenine will also, the composition of adenine will also be 15%. Okay, so because the adenine pairs with thymine, so both will have the same percentage. Okay, so total for both adenine and thymine, it is 30%. Okay, so to get the percentage for guanine and cytosine will be the rest of the 70%. Okay, the rest of what is left, okay, which is 70%. Okay, 70. So for both guanine and cytosine, you have to divide 70 by 2, okay, by 2. So obviously for guanine and cytosine, each will have 35%. Okay, 35%. All right, so if you add up this percentage, it will equal to 100%. All right. Okay, question 3. DNA is double-stranded while RNA is single-stranded. Give another structural difference between RNA and DNA. Okay, so you have to look at this question really carefully. Alright, in this question, it asks you to give the difference between DNA and RNA in terms of structural difference. So please highlight this. Okay, so for example, if you were to answer like, okay, DNA is found in the nucleus. Okay, RNA is found in both nucleus and also cytoplasm. Your facts is correct, but you are not answering the question, so your answer will be wrong. Okay, so you have to really take note. What does the question want? The question wants in terms of structural difference. So you have to look at the structure of the DNA and RNA. What is the difference between the structure of DNA and RNA? So obviously, as you can see from the diagram here, okay, you know that the obvious difference, okay, between DNA and RNA is that the molecule for RNA is smaller. Okay, RNA, the size is smaller or the size is shorter as compared to DNA, which is the size is longer and longer or larger. Okay, so that is one obvious difference between RNA and DNA. Okay, so you can erase this out to write the correct answer. Okay, erase it out. Okay, so DNA is the size is larger or longer. Okay, for RNA the size is smaller or shorter. Okay, okay, that is that one you can get, you can straight away get 1 mark. And then another difference that you can take note is that in terms of the base that are present in the nucleotide of these two nucleic acid, which is for DNA, the bases of that are found in DNA are adenine, guanine, cytosine, and thymine. And as for RNA, the base that are found in RNA, the nucleotide of RNA will be adenine, guanine, cytosine, and uracil. Okay, if for example you were to only write okay, in RNA there's presence of uracil, and in RNA there's presence of thymine. Okay, so your answer would also be accepted. Okay, because these three bases are considered as their similarity. Okay, and then another thing that you can also write for this answer as the one of the structural difference is in terms of the 5 carbon sugar. For DNA, the 5 carbon sugar will be deoxyribose. Okay, deoxyribose. And as for RNA, the 5 carbon sugar will be ribose. Or for the same mark, you could say that okay, for the 5 carbon sugar for DNA, at carbon number 2 of the 5 carbon sugar, there's presence of hydrogen. Okay, hydrogen at carbon number 2. And as for RNA, the 5 carbon sugar at carbon number 2, okay, there's presence of hydroxyl. Okay, hydroxyl group. All right. And then maybe you could also write as one of your answer. Okay, it already mentioned. Okay, DNA is double stranded, RNA is single stranded. Okay, and because okay, another answer that you could write. Okay, so because DNA is double stranded, there's presence of hydrogen bond. Okay, there's presence of hydrogen bond that is present between the two bases. Okay, the pairing of the two bases on the complementary strand. Okay, so you have the complementary strand here. Okay, adenine and thymine. So there's presence of 2 hydrogen bond. Okay, so always remember between adenine and thymine, there's presence of 2 hydrogen bonds. And as for guanine and cytosine, there's presence of 3 hydrogen bonds. So therefore, in DNA, there's presence of hydrogen bond that hold the bases together. And as for RNA, because the molecule is single-stranded, there's no pairing between the bases. So there's no hydrogen bond present in the RNA molecule. Okay, except for maybe tRNA. Okay, so tRNA is single-stranded. Okay, but the molecule of tRNA is actually folded into a clover leaf shape. So there's a segment of on the tRNA molecules that seems like it is double-stranded, but it is actually single-strand RNA molecule that fold into a clover leaf shapes. Okay. All right. Okay, so those are some of the structural difference that you can write for the answer. And another thing that you have to take note that whenever you answer question that involves you to give differences between two items, okay, in this case DNA and also RNA, so make sure you always compare the same thing. Okay, you have to compare apple to apple. Okay, for example, if you say that the size of DNA is larger, you also have to mention for RNA the size which is smaller. You cannot mention okay, DNA molecule is larger, whereas RNA there's presence of uracil. Both statement contains the right facts, but you are not comparing the same category. Okay. All right. So B, figure 6 shows the formation of a type of fat molecule which is K from. Okay. Figure 6 shows the formation of a type of fat molecule from K and L. Okay. So which is K here, as you know, it is actually, what is it? Glycerol. Okay, and L is actually fatty acid. Okay, so these two molecules will join together in a process we call it as esterification. Okay, esterification, where in the process it removes the hydroxyl, okay, from the fatty acid and the hydrogen from the glycerol. Okay, so the removal of hydroxyl and also hydrogen from the two molecule will remove water molecule. Okay, so because it involves the removal of 3 hydroxy group and 3 hydrogen atom from the glycerol, okay, respectively fatty acid and glycerol, so therefore 3 molecule of water will be removed in the process of producing M. So what is M? M is M is triglyceride. Okay, triglyceride. Okay, so draw the chemical structure of M. So you just simply join the two molecules together after you have removed the hydroxyl and also hydrogen atoms. Okay, so just join them together. Question B. Figure 6 shows the formation of a type of fat molecule from K and L. K, as you can see, it has three carbon atoms bonded to OH, which is hydroxyl group. Okay, so K is glycerol, which is a type of alcohol, and L is 3 molecules of fatty acid. In the process, the reaction that joins glycerol to 3 molecules of fatty acid is a dehydration reaction, or to be specific, because it involves the production of a fat molecule, the reaction is called as esterification. So esterification or dehydration. Why does the reaction called as a dehydration reaction or condensation? Is because it removes the process, or the reaction removes water molecules. Okay, 3 molecules of water. So for the fatty acid, it removes the OH here. Okay, and as for the glycerol, it removes the hydrogen from the hydroxyl group. So the removal of OH, hydroxyl, from fatty acid and hydrogen from the glycerol produces or discard 3 molecules of water. Okay, so the product will be M. Okay, so you join the glycerol and the fatty acid together to form M, which is triglyceride. Okay, so to produce 1 molecule of triglyceride requires 1 molecule of glycerol and 3 molecules of fatty acid. So figure 1 asks you to draw the chemical structure of M. So you just join the 2 molecules together. Sorry, you just join the 1 molecule of glycerol and 3 molecules of fatty acid together, where when you join the 2 molecules, the glycerol to fatty acids. Okay, so you have to join the oxygen to the carbon. Okay, so it is something like this. Okay, you just draw the molecule properly, where you have the hydrogen, okay, carbon, O. Okay, so O, you bond with the carbon of the fatty acid and the R group, okay, which is the hydrocarbons. O, C, double bond O, R, and then another carbon. Okay, hydrogen, the carbon, so you don't forget to add the hydrogen, and then oxygen, carbon, double bond with oxygen, and then the hydrocarbon tail there. Okay, so the hydrocarbons, name the bonds that so this is basically 1 molecule of triglyceride. So for 1 mark, you have to draw the molecule properly. If you get one atom wrong, okay, drawn wrongly, so you will lose 1 mark. So for example, if you forget, okay, to add oxygen here, okay, just forgetting 1 atom, so you will the molecule will be wrong and you will lose 1 mark. So please check your answer properly, okay, after you have answered your question. So please check. So question 2, name the bond that links the molecule K and L together. Okay, so all right, the bond that forms is between the oxygen, the carbon, and the oxygen here. Okay, the oxygen, carbon, oxygen, oxygen, carbon, oxygen. So the molecule involves the bond between oxygen, carbon, oxygen, which is ester bond. Okay, ester bond. So the name the bond that links molecule K and L is ester bond or ester linkage. Number 3, describe the solubility of M and the product of its hydrolysis in water. So M is a triglyceride. Okay, so triglyceride is a fat molecule, so obviously the fat molecule triglyceride will be insoluble in water. Okay, so that is 1 mark. Okay, the question worth 2 marks. So obviously any fat will be insoluble in water because the molecule is non-polar due to the presence of the hydrocarbon tail. So the hydrocarbon tail consists of many atoms of carbon bonded to hydrogen atoms. So because the bond between the carbon and hydrogen is already stable, there is no presence of charge or polarity in the hydrocarbon tail. There's no charge, there's no partial charge. Okay, so the molecule will be non-polar. Okay, so because it is non-polar, it is insoluble in water. So that is 1 mark. So that is M. Okay, describe the solubility of M and the product of its hydrolysis in water. Okay, that is for 1 mark. Okay, so M is insoluble. So that is the crucial the compulsory point for 1 mark. Okay, so you look at this question. It consists of 2 questions actually. First, you have to describe the solubility of M, and second is the solubility of the product of its hydrolysis. Triglyceride. Okay, so this is triglyceride. What is hydrolysis? Hydrolysis is the process where the molecule it undergoes a reaction that breaks the bond that requires water molecule. Okay, hydrolysis. Lysis means break. Hydro means that the reaction requires water molecule. So you want to break the triglyceride again. Okay, to produce back its component, which is fatty acid and glycerol. Okay, so for this one you can because the question is worth 2 mark, so 1 mark you have already mentioned for the solubility of M. Now you have to mention for the second mark is the solubility of its product, which is either you can choose you want to describe the solubility of glycerol, okay, or you want to describe the solubility of the fatty acid. You can choose which one. Okay, so for example, if you were to give the solubility of the glycerol, obviously glycerol, okay, is an alcohol because it has the hydroxyl group here. Okay, 3 groups of hydroxyl group, so it causes the glycerol to be soluble in water, or it is polar. Okay, it is soluble because it is polar. Okay, it is soluble in water because the molecule is polar. So that is 1 mark. So you get your 2 marks there. Or you can describe the solubility of the fatty acid. Okay, the component of the triglyceride. So the fatty acid would be insoluble. Okay, which is the same as the triglyceride is also insoluble. So fatty acid is insoluble in water because of the presence of the long hydrocarbon tail. Okay, so that is there is 3 possible point. You just mention for the second point, either if you want to describe for glycerol or fatty acid. M is compulsory. Okay, solubility of M is required for you to write, which is soluble. Number 4, suggest 2 reasons why aquatic mammals in cold regions stores lipid instead of carbohydrates. Okay, so example of aquatic animals that live in really extreme temperature, okay, in really cold area, whereas there's the surrounding temperature is really cold. This animal has to have a thick layer of fats, which we call it as blubber. Okay, for example, whale. The size of the whale is really big. Okay, the really big. So how can this really heavy animal able to swim in the water? Because it is due to the presence of the fat, the thick layer of fat. We call it as blubber. Okay, so the question, it says suggest two reason why aquatic mammals in cold region stores lipid instead of carbohydrate. So one of the function of the lipid is that lipid is less dense. Okay, it's less dense in water compared to carbohydrate, which is denser in water. So when they have a thick layer of fat, so this allows the animal to float, okay, or it aids in the buoyancy of that animal. Okay, so whale or walruses will have this fat to aid in its buoyancy. Okay, so that is for that. There's 2 marks for you. Okay, lipid is less dense compared to carbohydrate in water, which for the second point aids in its buoyancy. Okay, that is the 2 marks that you can get, or you can mention lipid produce more energy. Okay, lipid produce more energy compared to carbohydrate when the molecule or the lipid molecule is metabolically broken. So by metabolizing a lipid, so this produces more energy. So the animal requires more energy to warm its body. Okay, so that's why lipid is preferable, okay, compared to carbohydrate. And then another point that you can write is that lipid function as heat insulator, okay, to retain the body heat of these animals. So there are several points that you can get, so you can mention any of the 2 points.